Hello. How are you, everybody? Here we are again. I'm coming with uh, I'm coming with another topic on enzymology, of course, grade 11, unit 3, enzyme section. Today we shall see non-competitive inhibitors. Here, non-competitive inhibitors are inhibitors that do not have similar shape to the substrate, so that they do not complete for the active. I mean, compete for the active site. This is mistake, of course. Compete for the active sites. Instead, they temporarily bind to other sites away from the active site. Such sites are sometimes known as allosteric sites. And the binding of such inhibitors in the allosteric sites of enzyme produces changes in the conformation of the enzyme. You see, conformation of the enzyme, including the conformation of the shape of the active site, so that the substrate can no longer bind. You see, when allosteric inhibitors bind to the allosteric inhibitor site, they slightly change the shape of the enzyme, including the shape of the active site, so that the substrate cannot bind to the enzyme, which makes the enzyme inhibited temporarily, but in a non-competitive manner, because the substance, the inhibitor, is attaching not into the active site, but other site, special pocket, other site, which is known as allosteric site. Here, unlike the case in competitive inhibitor, the maximum rate of the reaction is determined by the amount of the inhibitors present, not by the amount of substrate. You see, in case of competitive inhibitors, yeah, the substrate uh, amount of the substrate uh, determines the degree of inhibition. Whereas in case of the non-competitive inhibitors, the amount of substrate matters. Thus, increasing the substrate concentration will not decrease the inhibition, unlike the case in competitive inhibitors. In competitive inhibitors, increasing the substrate, we have seen increasing the substrate has decreased the inhibition significantly. And the Vmax decreases while the Km stays the same. This is the opposite to the effect of competitive inhibitors. You can see here the graph. The first graph is without inhibition. The second, the Vmax value with inhibition. The third graph, Vmax value with more inhibition, Vmax value with yet more inhibition. What sort of inhibitor? The non-competitive inhibitors. As we increase the amount of non-competitive inhibitors, the Vmax keeps on decreasing. But the Km remains the same in this graph. You have also the other version of the graph, the Line-Weaver-Berg inhibition, decreased Vmax, increased 1 over Vmax, and the Km value is constant here. Example, suppose there are enough non-competitive inhibitors to bind to 70% of the enzymes. You see? Then 70% of the enzymes will be inhibited, irrespective of the substrate concentration. No matter whatever you increase the substrate, still 70% of the enzymes will be inhibited. That is, no matter how you keep on adding more and more substrates, still 70% of the enzymes will be inhibited, and the rate proceeds at 30% of the Vmax without inhibition. Note, such inhibitors are usually normal ways of controlling enzyme activities and controlling metabolic pathways in cells by using endogenous internal cellular metabolites as non-competitive inhibitors. You see, we have talked about the chemical properties of enzymes. One chemical property of enzymes is regulation. We have said the activity of enzymes is highly regulated and controlled. One way of controlling the activity of enzymes, if you don't need the product, is inhibiting their activities. One way of inhibition is by using non-competitive inhibitors, which are part of the metabolic product of the cell. Endogenous cellular metabolites, not something that comes from the outside in our food or drug that you put in your body. It is part of the chemical reaction process. Chemicals which are part of metabolic reactions in your cell, which attach on the allosteric site of enzymes, known as allosteric enzymes, and stop their activities temporarily, which is type of non-competitive inhibition. You see, therefore, non-competitive inhibitors are one way by which we control the activity of some enzymes known as allosteric enzymes by allosteric inhibition. You can see here one such example is one way this happens is by end product inhibition. Look, end product inhibition. Now, the end product is the chemical that is produced at the end of the pathway. And when the end product of a pathway allosterically attached on the first enzyme in the pathway and stop its activity. Which enzyme is stopped in the pathway? The first enzyme. So if the first enzyme is stopped, uh, all the other enzymes will not get substrate to convert. You see, you need to stop the activity of the first enzyme to stop the pathway. This can be described by this diagram. You can see here. The end product is E here. When the cell is not using E, E should not be produced, overproduced, and accumulate inside cells. So the E, the end product, attached on enzyme 1 in its allosteric site and inactivates it so that the whole pathway stops. The third, uncompetitive inhibition, the third type of reversible. Uncompetitive inhibitors bind exclusively to the enzyme substrate complex. I have told you this. They bind to ES complex, not the enzyme. So that therefore, enzyme substrate inhibitor complex, triple chemistry together will not give the product. This binding occurs after the substrate has attached to the enzyme, preventing the conversion of the substrate into product. As a result, the following effects are observed. You see now decrease in Vmax. Also, these types of inhibitors also decrease in Km. They decrease the Km value. This type of inhibition is rare but may occur in multi-substrate reactions. Multi-substrate reactions, enzymes which bind multiple substrates, that means many active sites. These are huge enzymes with many active sites, and such enzymes are usually known to be inhibited by uncompetitive inhibitors. You can see this graph, the graph with uncompetitive inhibitors. You see, both the Km and the Vmax are affected. What happened to the Vmax? Decreased, and what happened to the Km value? The uh, Km value has decreased. The same is the graph, Line-Weaver-Burk graph. And reversible inhibitors. Reversible inhibitors bind to enzymes tightly and permanently, usually by strong covalent bond, so that permanently altering the enzyme and then inactivating it permanently, of course. You can see the graph here. Without inhibitor, you can see competitive inhibitor there, it has reached. Non-competitive inhibition, uh, of course, then I think this graph talks about competitive and non-competitive inhibition. The reversible inhibition, both the Vmax, the Vmax is decreasing, as well the Km value also is increased. Application of enzyme inhibitors, huge area, as I've tried to tell you at the beginning of this presentation. Application of enzyme inhibitors. Enzyme inhibitors have got huge application in the medicine, in pharmacology. The knowledge about inhibitors has many applications in biochemistry, in medical science, pharmacology, in agronomy to prepare inhibitors designed to inhibit activity of some pests, for example, which can destroy our crop plants, so that we can save our crop plants, increase productivity. Therefore, application in biochemistry, in medicine, and agronomy. In biochemistry, used to gain knowledge about the shape of the active sites and the amino acid residues in the active site of enzymes to determine just what groups on the substrate are being recognized in the active site. This in biochemistry, when you are using enzyme inhibitors, it allows the biochemists to gain knowledge on the shape of the active site, how the active site is interacting, what amino acid sequences are there, specific amino acid sequences or types in the active site, and to determine just what groups on the substrates are being recognized in the active site. Of course, also it helps to study the chemistry of the substrate that are interacts with the active site. Used to gain knowledge or information about regulation or control of metabolic pathways, how to control, how regulate, control metabolic pathways. In medicine, used for designing drugs, including antibiotics, and also to design drugs in psychiatry, mental abnormalities, of course. The other is in agronomy, used to design insecticides or pesticides and herbicides, chemicals which inhibit enzymes of herbs so that the herbs will not grow and decrease productivity in agriculture. Thank you very much. This is the end of the second presentation, please. And I will come next time by another presentation on enzymes. And keep awake, work hard, and there will be plenty to eat, they say, please. Don't forget subscription, liking, and sharing my videos, and also put your comments underneath. Thank you very much. Ciao.